Hello everyone, I'm Denver, and this is my second talk of my scientific English report. My topic is diverse interaction between corals and coral killing sponge, the Terpius hodinota, and the author is these guys. So corals, we all know that corals are the power plant in our ocean environment. It produces a lot of energy for the uh, biological system in the ocean. So corals mostly lives in the tropical clear water, which the temperature is above 18 Celsius, and it's the main energy resource in the ocean environment, and it produces nutrient by undergo the photosynthesis throughout in the bodies. So the scientists found out that the coverage of the corals is decreasing, but they want to know why, because they found out that nearly 30% of the coverage is lost in Guam on the reefs, and also 30% of the coral coverage is lost on the 100-meter transect belt in Green Island. And we know that the corals are the power plants in the ocean system. And what if the power plant shut down? Means that the whole system will crash because they don't have enough energy and nutrient to live. So the scientists start to think about, are the corals sick? Sorry about that. Or is it that the corals got killed by other animals or so on? And they found out that they focused on the coral diseases. There are actually 29 kinds of coral diseases, like the Aspergillus and the white-pox disease and the black band disease. And these are like the coral diseases. It's common. I think that's the white pot one. So you guys can see these are the regular corals, and they got infected by the diseases and become white, and it's ugly, and it don't have enough energy to support the system. And like these are some examples of diseases. So actually, for coral diseases, it's specific infection for coral species. It means that for one disease can only affect one kind of corals, but and it's usually infected by bacteria, fungi, and protozoa. But there are one exceptions. They found out that one kind of coral diseases can affect a lot of different kind of corals, and it's found in 1993, and it's a kind of cyanobacteria sponge that was found. It's so strong that it can infect like most every kind of the corals in the ocean, and it was first widespread and in fact in Guam in 1973, then in that place till in Green Island in Taiwan in 2007, and it is called the coral killing sponge. The Kellen sponge is Terpius hoshinata, and this is the name of the formal name of this kind of sponge, and it's usually black and gray, has pin-shaped spicules. Spicule is the pointy tins on the end of the surface of the sponge, and this is the length of the spicule. It's actually 245 millimeters, and the wide is about 3 mm, and the head is about 5.6 mm. And the Terpios hoshinata, the sponge contains a high percentage of intercellular cyanobacteria. And it is also well known as the coral black disease. We all know that black disease is a really big disaster that happened in the mid-century. And it also grows on uh, Ceratophyllum and reefs. It's really weird because usually the sponge won't grow on these kind of area, and it even grows on dead corals. And this is the picture of the corals that was infected by this killing sponge. So you can see this should be the regular color, but it was infected by the gray and black sponge. So is on this picture. So it used to be a beautiful and colorful place, but it was infected, and it's since so deadly and poisoned. So the scientists start to study on what is the interaction between the corals and the sponge. So is it that the sponge will uh like in will put some toxic chemicals into the water and that infected the corals, or is it it occupied the whole plant? So in 1973, Brian, he found out the Terpios kill the coral killing sponge was also infected by the toxic chemicals. Means that they do came out with some weird chemical that is toxic for the corals, and they found out if it's next to a fungus corals, the tentacle will quickly retreat and die after 24 hours if the sponge is next to it. And the terpios moves forward by later propagation, propagations, extending short, fine tendrils across previous to new substrates. And Terpios hoshinata can develop tissue and threads instead of the whole sheet of tissue to move over the shaded area and establish a new territory. So they start to do their field observations. In Green Island, they start their observation on August in 2008 to till July in 2010, and in Gomgon, two spot in Gomgon and Chaiko, and it was recorded by underwater camera by while they do the scuba diving from 3 to 5 meters in depth on 28th July on 2010. And then they put the picture examined by the SEM, also known as the scanning electron microscope. So this is the Gongom spot, and this is the Chaiko spot. So on. Then in Green Island, they found there are 19 species of scalar raised TNT and corals and one kind of hydrozone corals. They samples from two different colonies, and the Terpios coral specimens were sealed in plastic bag underwater and collected and preserved in fixation once the diver had left the water. So the first kind of reaction between the corals and the killing sponge is the hairy tips, and it was found to occupied by cyanide bacteria and the sponge tissue and spicules. So this is the spicules of the sponge. It's the pointy, tiny tin on the end of the surface. So in these four pictures, the first one is the picture that they took under the water, and this is the hairy tips. This is the normal coral surface. And these three pictures are the SEM scan when they do after they go back to the lab. And for these pointy stuff, it's the spicule of the sponge. And this one, and these. And it's disgusting. And the second and third one, it's the tick tissue. What is that? Oh, okay. And the thick tissue thread on it's this one. And the thick tissue threads were an extension of sponge tissue. So this is the coral surface, and this is the sponge tissue, and it will extend its tissue to affect the corals. And these two are the these two are the SEM pictures of this picture, and this is the terpios front part, means the sponge part, and this is the spicules of the sponge, and this is the coral surface. So you can see it's still black and gray and infected, the beauty and the colorful and the nutrient of the corals. And the fourth one is the disintegrated tissue. So you can see this is the normal corals, and this is the black disease. It's really disgusting. It's ruined the whole environment. And these are the tissue part, the terpios, and this is the corals and the spicules. It's too hard to describe. And this is the form that after they go back to the lab. So there are 20 kind of corals, and these four are the impacts, the interaction between these. And the positive part means that there is this kind of interaction, and for the negative part means there are no this kind of interaction. And so this is the resort. 13 out of 20 have hair tips, and 7 out of 20 have the thick tissue threads, and 12 out of 20 have compact edges, and 1 out of 20 have the distinguated tissue. So hair tips and the compact edges were the two most common features in the interactions. And features of the border between the two antagonists were not uniform among coral species. And the interaction between the corals and the killing sponge are dynamic and also not species to specific. Means that I mentioned most of the diseases will be species to species, but this kind of sponge can infect all kinds of corals. And this is the end of my speech. Thank you.